What is cross-pollination? It's when a plant is pollinated with the pollen from another plant. We can simulate that natural process in order to get specific variations of a desired plant, in this case, tomatoes. Before we delve into our discussion on cross-pollination, we must first understand the anatomy of the tomato flower. A tomato plant has perfect flowers, meaning they have both female and male reproductive parts. The female part, called the pistil, consists of the stigma, style, ovary, and ovule. We are concerned with the stigma, for it is the part of the pistil where the pollen is germinated. The male reproductive part is known as the stamen and consists of the anther and filament. The anther is where the pollen is produced. Let's think about how natural pollination happens. The gentle vibration that comes from a bee landing on a plant shakes pollen off the stamen and onto the bee. We replicate that process by using this electric toothbrush to shake the pollen from the mature male flower into a capsule. It is important the male flower be bright and yellow. If you look closely inside the capsule, you can see the pollen we have successfully collected. Now on to the female flower. As you can see, the petals have not opened yet. We need a young female flower to ensure that self-pollination has not already occurred. To prep the female flower for pollination, we remove the outer green sepals, inner petals, and the anther with sterilized tweezers. All that is left is the exposed stigma. We then gently add the collected pollen from the male flower onto the exposed female stigma, ensuring an adequate amount of pollen is being transferred. Crossing tags are always labeled with the female parent name first, crossed with the male parent name second. On the back of the tag, we put our initials to show who made the cross and the date. We use a slip knot to put the tag only around the bud that we pollinated. Throughout this whole process, it's important to remember to properly sterilize equipment with isopropyl alcohol to avoid cross-contamination. We also have to carefully label collected samples. This allows us to know exactly where the pollen came from and which flowers have been cross-pollinated. And voila! We have successfully cross-pollinated this tomato plant. Fertilization has occurred, and in about 45 to 60 days, our little flower will have grown into a hardy tomato.